This is episode 314 of the Stem Cell Podcast, ISSCR 2026, with Doctors Fiona Doetsch and Nozomu Yachie. Hello, everybody. Back again. It's Daylon James and Arun Sharma. Welcome to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotech supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on future guests. Today, we have a couple of guests with us to discuss the biggest meeting of the year, ISSCR 2026 annual meeting. That's Doctors Fiona Doetsch and Nozomu Yachie. They're here to talk about the upcoming annual meeting, which is happening in Montreal between July 8th and the 11th. We've also got our usual roundup of recent highlights and stem cell news. That's coming up in just a sec.
But first, registration and abstracts are open for the Global Stem Cell event, the ISSCR 2026 annual meeting, July 8th to 11th in Montreal, Canada. Join nearly 3,500 scientists, clinicians, and industry leaders for 4 days at the frontiers of stem cell science, regenerative medicine, and engineering biology. From fundamental discovery to clinical breakthroughs, this is where collaborations start and new ideas take shape. Submit your abstract by February 25th for a chance to present your work and be considered for travel and merit awards, and design your dream session and submit a science spotlight proposal by February 25th as well. Be part of the global conversation shaping the future of stem cell research. Visit isscr2026.org/scientific-topics, where you can sort the agenda by theme. Topics include bioengineering, organoids, neural stem cells, aging, and more. And indeed, the first paper we're going to talk about here in the roundup today is an organoid paper, and organoids are going to be well represented at ISSCR 2026 in Montreal. This is a unique application of cholangiocyte organoids coming from the lab of Alan Ka-Lun Chung over there in Hong Kong, China, Department of Biology at Hong Kong Baptist University. First author here is Huodong Jie. So this is using organoid models to understand the infection mechanisms of human cytomegalovirus, or HCMV. And HCMV is associated with different disorders of the bile duct in both immunocompetent and also immunocompromised individuals. And these disorders can include sclerosing cholangitis, different biliary structure abnormalities, AIDS, cholangiopathy, biliary atresia, and some post-transplant issues as well, including different biliary complications that they refer to here. And so there's a lot of these clinical associations here with this human cytomegalovirus and these different biliary disorders, but in spite of all that, it's still not really well known whether this virus, HCMV, can actually directly infect the bile duct epithelium and actually cause these downstream pathogeneses. And so the study itself is sort of straightforward. They created an iPS-derived cholangiocyte-like cell, CLC organoid model that ultimately was used for an infection model, right? So they wanted to show if indeed HCMV could infect these cholangiocyte organoids, and what would happen if that was the case. So the first thing they showed was, yes, indeed, that the HCMV was able to infect the organoids and actually reduced the overall growth of the organoids, changes in their structure as well, deformations in their overall morphology, and importantly, a loss of their barrier integrity and barrier function. Then they did a deeper dive into the transcriptomics, and the bulk RNA sequencing showed that the HCMV infected organoids actually had a pretty highly enriched expression of pathways and genes involved in EMT, epithelial to mesenchymal transition. And then they figured out a way to alleviate some of these infection phenotypes by a blockade of TGF-beta signaling, which is actually able to abrogate the EMT induction effect in the HCMV infected CLC organoids. And then they took an even deeper dive with single cell RNA-seq, which makes sense. These are organoid models, so single cells pretty well suited to transcriptional analysis at the single cell level of organoids. And the single cell revealed that the HCMV infection actually happens in different clusters, different cell types in the organoids with actually distinct viral and host gene expression profiles. And again, TGF-beta popped up here again, as well as different genes related to EMT. Then while organoid models and iPS-derived models are great, of course, we know they're not perfect. They have certain immaturities. So they use the gold standard, actually, and they confirm these findings in primary patient samples in patient liver tissues, actually, by examining the expression of these HCMV DNA positive cells. So they're actually finding populations of these cells that incorporate the HCMV infection, and the same signaling mechanisms were altered in the primary tissues as well. So that's a really cool way to correlate the studies and the mechanisms that you're identifying in the IPS model, and a way to confirm that what you're seeing is not just an artifact of the IPS system itself. So they ultimately found two modes of infection by the HCMV virus in these CLC organoids, which has both a productive and a latent feature. So it's kind of two sides of the coin here. And ultimately, I think it's a useful model system for studying infection mechanisms. And we've talked about this before years back when COVID was all the rage. Everybody's making your favorite organoid type and different iPS-derived cell types and infecting them with the SARS-CoV-2 virus. For one, just to see the infectivity and the tropism of the virus to different cell types. And this is kind of an offshoot of that in a way. It's using these organoid models and these cholangiocyte organoid models to understand infection mechanisms of this really interesting virus, HCMV. So cool model system, and I think the downstream applications for other types of viruses are pretty obvious here.
I was thinking exactly that when you were talking about this, and when I had a little look before the show, that this really reminded me of the heyday, right? Where it was COVID and Zika before that, remember? And that really was, just as you said, just about, okay, let's take the whole panel and figure out what the tropisms are. And maybe we'll get at some elements. It seems like it's a vascular disease, et cetera, at least in the case of COVID. But this, I think those had the benefit of having tremendous interest and demand, right? You could publish a lot of things that probably wouldn't go through now, just based on the critical, DEFCON 1 that we were at. But this, I think, is highlighting the most exciting application of organoids in this space that we haven't seen so much before. And we have seen it to some degree, but I really enjoy this because it takes the kind of model system out of the animal, right? It's very much in keeping with these NAMs that we've been talking about and we'll continue to talk about at the ISSCR. It makes this accessible, ex vivo model that has, I don't want to say perfect fidelity, but some fidelity to the physiological ground truth, right? But then it unpacks this cellular basis and the etiologies of disease and making those links, which I have always thought is the most exciting application of organoids, at least when you're talking about infection and the cellular sequelae thereof, because you can see it in real time, whereas before you just have the associations, right? You infect the mouse or you look in the patient and you see the downstream effects, but here in real time, right? With high granularity, with the seq and all that, you can really deconstruct the cellular basis of pathophysiology here. And I love that. And I have a similar kind of story, although totally in a different direction, but similarly taking the system out of the body. And in the context of disease and infectious disease in particular. My story is about, liver and hepatitis in this case, but a similar idea of like using these systems. That's not a stem cell or organoid story at all, I should disclaim. But we'll get to that. But this one is, and it's a story about using this, I think, in the best way, in the way that we all saw the promise to tackle infectious disease in the cross section with cellular sequelae. So very cool story for me.
Yeah, I also liked it as somebody who did a little bit of a COVID virus work in around 2020 and 2021 myself. I really liked it because they correlated their IPS findings to the primary tissue. I mean, there were a lot of studies out there during COVID times where they were infecting whatever IPS or artificial cell type with the SARS-CoV-2, but they weren't really doing the gold standard of, actually seeing if it's actually truly clinically relevant and that that cell type that they're looking at in vitro is actually being infected in situations of COVID-19. And I'll give the example of the cardiac biology, right? And our lab showed that isolated iPS cardiomyocytes could be infected by the SARS-CoV-2 virus, but the gold standard was truly endomyocardial biopsy, getting a primary sample of someone's heart in the situation of COVID. And we and others showed that indeed, if you could use electron microscopy, you can find the COVID virus particles in the myocardium in situations of really severe COVID. So that was a way to correlate one-to-one our in vitro iPS-derived data with true clinical data. So, and like I said, I'm a big fan of the fact that they did something similar here as well. So the models are great, as we've talked about, but you gotta make sure that they have clinical relevance in a way, and especially when studying infection mechanisms like this. So I'm happy they sprinkled that on the top here.
No, for sure. That correlation's strong. And they even concede at the end that there is still much work to be done to link the cytomegalovirus to biliary atresia. There are some kind of disconnects looking at congenital. They say it's not really a manifestation thereof in the biliary atresia. But as you said, as we've been discussing, I think extending beyond just the correlation, but causality and beyond. So a really fun story. As I kind of teased, I got this story about the liver, and it's a xeno. It's a xeno story. You know me. You know us. If there's anything xeno out there, we're on it. And this is a nature medicine story. I also like to highlight once these kind of cross the Rubicon into clinical practice and nature medicine level, which means that they're on some level showing maybe clinical trial results, which we've reported before here, is just a really exciting, fun, experimental approach here with 4 brain dead patients. And the hook here is using extracorporeal liver cross circulation, right? So there's all kinds of liver failure. We talk maybe mostly about like degenerative or chronic liver failure. You think of like alcoholism, but there's also acute is probably the number one cause of liver failure. Overdose of aspirin, Tylenol, for example, due to toxins. Also viral infection, hepatitis. Get your vaccine for hepatitis B. I know a lot of resistance there, but the damage really outweighs the cost of the jab. Doing my little PSA. Totally unnecessary, probably inappropriate, but there we are. But getting back to the story, whether you have acute or acute on chronic, which is that you have like an infection that exacerbates some pre-existing chronic liver damage due to maybe hepatitis or alcoholism, you get this impaired hepatic metabolism and toxin accumulation, right? And the stats here are pretty jarring. Ultimately, this results in multi-organ failure for a ton of patients with a 4-week mortality rate of around a third. A third of these patients are going to die within a month. So this is pretty big in terms of the impact. And the reason there for such a high mortality is because you can get a liver transplant, right? Of course, that's effective. But the presentation for a lot of these patients makes them unsuitable for transplantation. Of course, there's also a huge shortage, right? Of course. So that leads to the fact that fewer than 10% of these patients that have this acute on chronic liver failure, less than 10% of them undergo this emergency transplantation. So we need a bridge system, right? That can either take the patient to the point where they can receive a transplant, one becomes available, or, oftentimes you can just keep this patient going until their own liver recovers. We talk about it, right? The liver is remarkably regenerative. So if it can recover to the point where it has a threshold of native liver function, then patient walks out the door. So you can get these support devices that are currently out there, similar to the one I'm talking about, but they remove. It's kind of like dialysis. They remove toxins and they modulate inflammation. But when you run the numbers, there's no clear survival benefit in these approaches versus other approaches, and it's because they don't provide that comprehensive metabolic support that a native liver could provide. Now, there's the cell-based therapies, all right? We talk about hepatocyte clusters, which I think hold a lot of promise. But to date, they have not reached the level where they can substitute for transplant liver, or even these extracorporeal like dialysis like approaches. So that brings us to the xenogenic livers, right? We've talked about this before a lot with these triple glycan knockouts and many different companies approaching this way of providing an unlimited or as limited as there are pigs, which we know in America, they're more than enough, unlimited organs for transplantation. And there's been, I think, exciting degrees of success, but of course, some setbacks in terms of immune reactivity. So we're not quite there yet. And this is offering a kind of alternative or kind of complementary, maybe, approach, which is this extracorporeal liver cross circulation, which has several advantages, in particular in the context of this temporary bridge support, right? So one, much less invasive. You only need a venous cannulation, right? So this is, I wouldn't say minimally invasive, but you don't have to, open the patient. And you can continuously monitor the xenograft function outside the body. Like it's right there. I don't want to say it's like an organoid, but it's available, accessible. And also on that same note, like if the problem does arise, you can discontinue the support immediately without having to have, of course, any surgical intervention, right? So for those reasons, this group, which was a real powerhouse, big list of authors, led by Abraham Shaked and Alex Sager, Peter Rees, and Peter Friend. So Abraham Shaked and Peter Friend are from the Penn Transplant Unit, and Alex Sager and Peter Rees are from Oxford. And Peter Rees is associated with this Organox, which is this pig, organ glycan knockout thing, associated organ ox from Oxford. You get it. Anyway, that group of individuals, as in addition to a number of collaborators, pioneered this approach of using the extracorporeal liver platform to evaluate. And this was kind of what the point of the pilot is, that they wanted to see what the immune response was, in addition to like whether or not the liver could provide physiological support in a brain dead human model. And that's the key here, right? All these first in human trials have been done in decedent models. And in this case, they had 4 human decedents. They were administered immunosuppression. I think that's an important point. In the first 3 of those, they had this extracorporeal liver circulation for 3 to 3.5 days, with the native liver remaining in situ. And then the 4th one, they actually hepatectomized the decedent and then followed that with 48 hours of the xenogenic liver support using the ELC, this extracorporeal liver circulation. What they found was that these livers, they biopsied them after, the extracorporeal ones, found that they preserved the parenchymal architecture, but they did have a mild immune infiltration and IgM deposition. So the similar kind of immune reaction infiltration that we've seen before in the transplant patients in the case of kidney, heart, lung, liver as well. Uh, but the livers produce bile. They supplemented the native hepatocellular function, and they maintained in the fourth patient where there was no native liver, they maintained hemodynamic stability, normal pH, lactate, ammonia, and normalized ratios and sustained metabolic function. So I don't know. I'm sure there may be some challenges here that are probably aligned with the same challenge of transplanting these things in vivo. But I feel like this is not just for patients awaiting liver transplant, but kind of like symbolically as a bridge to the xenotransplantation that's going into the patients. I love the liver as this extracorporeal device, and you could see it. I don't know about scaling, but like dialysis is a thing. And just, I mean, I'm picturing like live liver dialysis centers that do this ELC could relieve a tremendous burden on the healthcare system in a pretty large proportion of patients that present both with this acute on chronic as well as acute liver failure. I mean, I could see this being in the bag of every ED, obviously consultation with surgery, but like still, I think this is maybe a new entry in terms of modality for this kind of semi transplant therapeutic based on xeno origins.
Yeah, we love the Xeno here. Every time there's a Xeno paper, high profile Xeno paper, it feels like we gotta cover it. And I think it's important that you mentioned that indeed this is a bridge. This is not a permanent solution. This is a bridge until you have a true liver replacement available for long-term integration. And that's the case with the other organ types as well, like the cardiac situation. It's initially intended as a bridge. Um, and it's a big step forward, especially with that patient four with the hepatectomy. But like you alluded to, it's not perfect. You still have the immune infiltration, and I think these triple glycan knockouts are really impressive, but it's not a magic bullet. There's still more that has to be done here when it comes to the immunomodulation. So I'm excited for the progress, especially over the last 30 years. I can't even imagine us getting to this point 30 years ago, but part of it is thanks to advances in genome editing and all that enabled the modification of these immune genes. But, we still got some ways to go here.
No, for sure. And you wonder, I mean, like one idea that I was thinking here is, oh, we could extend this perhaps if, further using IPS derived cells or HPSC derived cells that are like mini liver type organoids. Only challenge there is that you lack the circulation, right? So the idea of cannulization dialysis doesn't really fit with that. But I do think that, the way things are going, that there is this next type function that this introduces, or next type modality that this introduces, which is the extracorporeal device, right? Extracorporeal organ, not of your heart, although maybe, I don't know, someday, but like kidney, liver, people carry around oxygen tank. Maybe they'd be carrying around their detoxifier or filtration devices for their blood. It seems far-fetched, but I think that incrementally we could approach something that's scalable. And I mean, it's kind of silly people walking around with their livers outside their body, but for sure, I think in a acute setting, inpatient, that this would certainly supplement our healthcare system in the future. I think there's a realistic possibility there that this could be the entry point for some of these stem cell based products.
Yeah, definitely a possibility. And you joke about the extracorporeal cardiac side of things. I mean, ECMO already exists, as we know, which is, extracorporeal membrane oxygenation, which is basically having an external heart just following you, what that you can walk around with outside your body. Um, and that's been a mainstay for heart failure. And talking to my cardiologist wife, she says that you can keep anybody alive on ECMO, it seems like, just, keep their bloodstream oxygenated and all that. So yeah, but we're talking about more the cell-based approaches here, and whether it's a combination of IPS or Xeno, I think the end goal is the same. So it is definitely a very exciting time for these large organ replacement and bridge studies, and I'm excited for the next few years as well. Shifting to something totally different, probably a little bit more traditional in terms of the things that we cover here. This is a cell stem cell paper focusing on intestinal stem cells, and certainly we have had a chance to talk to Hans Clevers, the intestinal stem cell guru himself. But this is a unique angle to the intestinal stem cell story, and this is actually talking about how sleep disturbance can trigger an aberrant activation of different vagus circuitry and ultimately induce intestinal stem cell dysfunction. So connecting sleep to the gut, who would have thought? Sleep disturbances, unfortunately, we all kind of deal with them, right? As parents, I'm definitely going through sleep disturbances right now as my kids have infected me with who knows how many different types of viruses over the last few weeks. I wish I got more sleep, that's for sure. And sleep disturbances are more than just an annoyance, right? They can sadly lead to more serious chronic disorders, unfortunately for me, I guess. Um, so, for example, chronic gastrointestinal diseases, that's something that sleep disturbances have been linked to, including cardiovascular disease as well, but that's not really the focus here. So, at least when it comes to the GI diseases, that mechanism of how sleep disruption manifests in those GI diseases is not well understood, right? So it's some connection with the neural and the gut, from the brain to the gut, but that mechanism is still something that folks are trying to figure out. So here they're showing that in a mouse model, of course, they're showing that an acute sleep deprivation can impair intestinal stem cell function, leading to a shortening of the classic crypt-villus architecture and loss of Paneth cells as well. So they identify the dorsal motor nucleus of vagus, or DMV, as the sleep deprivation sensitive hub for the central nervous system that actually can transmit those sleep deprivation effects to the gut. So there's the mechanistic connection right there. Sleep deprivation, they're showing, is actually able to activate these DMV neurons, ultimately driving excessive acetylcholine release from the vagus nerve and into the gut, leading to some of that disruption. And they took a deeper dive into the chemical mechanism here and actually saw that the acetylcholine can trigger 5-HT release by enterochromaffin cells, and also suppress its reuptake through muscarinic receptors. So ultimately, there's a spike in this 5-HT, 5-hydroxytryptamine levels. And then the downstream of that is the elevation of the 5-HT can actually induce excessive oxidative stress. So that's ultimately what negatively impacts the intestinal stem cells through a receptor HTR4, and that is leading to further gut pathologies. So this was intestinal stem cell specific, and I'm sure there's other versions of the story that are very gut microbiome focused, because certainly that's all the rage these days, as well as figuring out the influence of various environmental situations on the gut microbiome. They didn't really dive into that here, but maybe there's a connection there as well. But ultimately, they're showing this unique sleep deprivation responsive neural circuit that can control intestinal stem cells, their viability, their health. And they also, by figuring out this intricate mechanism, linking 5-HT to oxidative stress, they also identify some potential therapeutic strategies for maybe mitigating this sleep deprivation related forms of gut disease. But for me, it's just, the easiest way to mitigate is just get more sleep, but easier said than done, I guess.
I'll tell you, I should collaborate with these guys. I got an idea for rescue, and that's a bacon, egg, and cheese sandwich. Late night, that does it for me.
Love it.
Rescue. Anyway, yeah, this is so deep. What a mechanistic journey, right? And this is why I don't want to. I know everyone's all gaga about these NAMs, but we talked to Joe about it, and he was very clear. We cannot bypass or do away with the animal models, and it's stories like this that really show you the value of applying this and integrating it, right? All the way from the brain to the gut, which is connections that are well established, right? So I don't think the hypothesis is out of this, crazy or anything. It's not a crazy revelation, but such a neat dissection of the links. And yeah, mechanistic in a way where, notwithstanding my rescue of fatty foods, um, it actually has a means of maybe addressing it. Although, as you said, I feel like just go back to sleep is probably the key. But I don't know, talk about military. It doesn't really even matter the applications, whether or not we're going to come up with the magic pill where you won't screw up your gut when you have an all-nighter, like, is less important to me than the understanding, right? And the mechanistic dissection and deconstruction here is just such a fun puzzle that they teased out here. So very cool story for me.
Yeah, and I think you make the military analogy. I think a lot of people can tolerate sleep deprivation, acute sleep deprivation for a while. I mean, ultimately, it does lead to significant issues, and I think sadly, this is the reality of medical professionals. And
Mm.
you know, as you know, sleep deprivation is the norm during clinical training. Acute sleep deprivation, chronic, whatever, and it manifests in a bunch of different ways, including certainly GI issues. So I think that there's a lot of potential observations and correlations that can be made here. One other thing that I thought was interesting, though, I mean, we talk about the utility of NAMS and Joe's emphasis on still using the in vivo models. One thought here is that, aren't rodents and mice nocturnal, right? So is that kind of a confounding variable in itself in the context of this paper? I mean, they can have very different sleep cycles from the rest of us, higher vertebrates, primates, whatever. So I don't know. Maybe that I'm sure they thought about that and were able to rectify it and control it in some way. But that is something that came to mind.
Now you got an idea for the next paper. We're going to have to come up with a super assembloid that goes from the brain all the way down to the gut. I mean, I wouldn't put it past some people. Serge, you get after it, brother. Moving on to the blood. I mean, the blood, you said it. This is kind of the tip of the iceberg in your story about how the sleep disturbance probably affects multiple compartments, but there's just a really well established link in the gut that can be dissected. The blood, probably, and other niches as well. So that's my segue. And this is a story really about the hematogenesis, right? Which is, of course, one of the holy grails of the field generally, stem cell biology, not just hematology. But looking at, and this has been studied as much as anything, the origins of hematopoietic stem cells. And there is still a lot of controversy surrounding it because the goal is so important, right? The fetal liver, though, and in fact, the primordia, the fetal liver, has emerged as like this fundamental hub, right? Where you get this heterogeneous set of hematopoietic progenitors that initially arrive from the yolk sac, and this is all in mouse between days 7 through 9. And then just after that, around embryonic day 11.5, the fetal liver is also colonized by these circulating multipotent progenitors that arise from endothelial to hematopoietic transition from the walls of these large intraembryonic arterial vessels, right? Like the AGM. And lineage tracing has shown that those intraembryonic derived progenitors they mature and then ultimately seed all the layers of the definitive hematopoietic hierarchy. And hematopoietic stem cells, right? Those repopulating cells, they also undergo active proliferation and expansion. Although, like for the most part, they're poised to differentiate and become all the downstream derivatives. Those stem cells, for a couple of days later, between 12.5 and 14.5, then the actual stem cells will expand, right? And then after this, you get this migration of these stem cells as well as progenitors throughout the whole body. But stem cells go and they hone into the bone marrow and the spleen, right? And so the fetal liver at multiple stages, from the yolk sac, this enteroembryonic, this expansion period, and ultimately the exit as well. The fetal liver microenvironment is a critical signaling hub. It's like a launch pad, right? Um, that instructs maturation, self-renewal, and differentiation of these multiple waves of progenitors and stem cells um, that ultimately sustain hematopoiesis for the rest of that animal's life. Now, we know so much about the niches for hematopoiesis in the adult bone marrow, endosteal, endothelial, vascular. I mean, there's been like literal street fights about the niche, hematopoietic niche at some of these meetings. Um, and they've got a lot of attention, but the bone marrow is kind of different. Hematopoietic stem cells in the bone marrow are relatively quiescent, and they only enter cell cycle and like in response to challenge or very sporadically at steady state. And on the other hand, the hematopoietic niche within the fetal liver remains relatively understudied. And that's where the group led by Cesar Nombela Arrieta, who's at the University Hospital and University of Zurich, um, they led an effort which, straightforward, bare bones approach, high tech, low tech, all of it. 3D microscopy, bulk RNA seq, flow, mouse transgenic models. They combine all these tools to map the dynamics in space and time of these niche cells and the hematopoietic stem cells that they foster during fetal liver development. And what they found is that it's at the peak, at least, there's these hepatoblasts, endothelial cells, and mesenchymal stromal cells. So very similar to the niches that exist in the adult, that they all express these pro-hematopoietic cytokines across the entire fetal liver. But just because the fetal liver is made up mostly of hepatoblasts, those were the kind of dominant cell type. And they regulated through abundant expression of niche factors, hematopoietic stem cell expansion, erythropoiesis, and mainly through KIT ligand, which is well established pro-hematopoietic ligand. And this was the cool thing to me, is that there's this cellular remodeling that takes place in the fetal liver, or becomes more epithelial, that's necessary for its maturation and like when it becomes endowed with its actual liver specific function. Um, and that remodeling actually coincides with the hepatoblast, the resident hepatoblast differentiate to undergo that remodeling. And that kind of is like a get out of here signal. It leads to the downregulation of all these supportive cues and coincides with the exit of these hematopoietic stem cells as they go migrate to the peripheral tissues, mainly the marrow and the spleen. So I like this story because it's been a long time coming. So many conversations about the adult niche and the fetal liver and yolk sac and AGM. All these niches have been appreciated and deconstructed, but I like this for its kind of longitudinal, although it is a short space in time. But so much happens in that time. This spatiotemporal mapping, I think, across the entire fetal liver is a really key piece of the puzzle. That's why it's a resource here in developmental cell. And I think that this will be useful, again, not just even for the practical application. There's a lot of stuff that we already knew. There's a lot of similarities to findings like in the adult. But I think having this resource nailed down and also just, again, how they puzzled it out was a really fun read. So I recommend it highly. Arun, what's your take?
Yeah, great story. It's a classic dev cell kind of story, and we love dev cell. Really beautiful, intricate analysis of different lineage pathways and so on, and amazing imaging as well. I think they did a good deep dive. I mean, they mentioned that they're they focus pretty heavily on the remodeling of the stromal components, and particularly the role of the hepatoblasts on the HSC expansion, but they didn't take a deep dive into some of the other cell types as well, like, the relevance of the MSC compartment. And they actually mention it here, different MSC subpopulations other than the stellate cells have been described in fetal liver, and these are including like the NG2 portal cells, a bunch of other cell types, mesothelial cells as well. So I think they still have some work to do here for those adjacent cell types and auxiliary cell types as well, and seeing how they influence this process. But nonetheless, really a great start here.
Yeah, and I mean, I feel like it's not going to be long before we see one of these kind of xenium maps or something that's much more comprehensive and granular. There was a lot of bulk seek in this story, which is part of me. I mean, all of me loves that. I love it when you take tech that's very well established and robust, and maybe not give you the same level of resolution as the new tech, but if applied carefully and deliberately can really provide a vast and valuable resource. And I think that this is going to be a great reference point for all those future, much higher resolution maps, because it gives specific landmarks in space and time that can then be focused on at a higher level of detail. So, again, a nice story, a nice uh, ground truth to build upon. And now moving on to our conversation with the leadership at the ISSCR about the annual meeting in Montreal. But before we get there, I have a quick message from STEMCELL Technologies. Are you interested in exploring the applications of organoid cultures? You ought to be after this episode, after listening to any one of these episodes for that matter. You should get a free wall chart showing how organoids are used as model systems to study infectious diseases, apropos to this episode, cancer, congenital disorders, and tissue regeneration. To learn more, visit stemcell.com/organoids-modeling-wall chart. All right, you guys, as I said in the open, we're coming around to that time of year. And as we have done the last few years, we have a special interview with some of the leadership of the ISSCR. And on today's show, we're going to talk with Dr. Fiona Doetsch and Dr. Nozomu Yachie. But first, I want to give a bit of information about the ISSCR 2026 annual meeting, which is fast approaching. The dates are July 8th to the 11th. The location is Montreal, Canada. And the venue, which I'm going to struggle with the pronunciation, so forgive me, is the Palais des Congrès de Montreal. I'm sure I screwed that up, but look it up. It's kind of phonetic the way I did it. And the co-sponsor is STEMCELL Technologies, who also, disclaimer, runs this show here. You can get more information at www.isscr2026.org. And just a bit about the ISSCR annual meeting, if you've been living under a rock, or if you're new, which makes you even more welcome. This is a big meeting, guys. It brings together global leaders across all types of science, basic, clinical, also technology development, translational research, and of course, with the focus all years, but in particular in this year on the future of clinical application of these therapies. And I think some of the highlights for me as I'm looking through the packet here is that more than 75% of the invited speakers are first-time annual meeting speakers this year. So that's really a reflection of the deliberate effort to bring in new voices, new disciplines, and perspectives from all around the world. And that's really the big picture idea of the ISSCR annual meeting. And this year, it's built around the themes of integration and convergence. And this is all stuff we've been talking about on this show for a long time now. With the state of the art in science, we're talking about bridging model organisms to humans, both in vitro and in vivo. We just talked a few episodes ago about these new approach methodologies that are a point of emphasis now with the NIH as well as the ISSCR. We talked about that with Joe Zhu. We're going to talk about that again in our next episode. And also bridging discovery and clinical translation. And of course, the program is always tech forward. But in particular, I think the value of the ISSCR annual meeting program is that while it is tech forward, it's always anchored in the biology and the physiology. And that's where we're looking for innovation. And this year, highlighting advances in computation, imaging, bioengineering, and of course, AI, ubiquitous AI, and how those different specialties and tools are transforming the questions that scientists can now ask. So that's a little bit of the run up to the meeting, a capsule, if you will. Now let's get to the speakers, because our interviewees, because they're gonna tell us more, and that's what we're really here to listen to. First, Fiona Doetsch, who's head of a research group, also professor at the University of Basel, also vice president of the ISSCR. So we got some big guns here amongst the leadership. She, along with Nozomu Yachie, are co-chairs of the program committee of the annual meeting this year. And her research group investigates stem cells in the adult mammalian brain. The lab's aim is to define the identity, lineages, and regulation of adult neural stem cells, elucidating the molecular and cellular pathways underlying their regulation, which could provide insight into brain repair. Also, as I said, Dr. Nozomu Yachie, who's co-chair of that program committee of the annual meeting, who is professor and research director of the School of Biomedical Engineering at the University of British Columbia. The Yachie Lab is developing DNA event recording tech, which I love. I mean, I read that Nature Biotech paper that he put out, and it's one of those things, you know, paradigm shifting tech, and reflective of kind of what we're talking about at the ISSCR, not in whole, but in part this year at the meeting. But these DNA event recording technologies, they enable high-resolution molecular and cellular information of individual cells in a multicellular organism to be progressively stored in these cell-embedded synthetic DNA tapes. Real future tech there. So yes, those are our guests today. After that long preamble, welcome you guys, and thank you so much for joining us.
Thank you so much for having us. Delighted to be here.
Yeah, thank you very much.
Yeah, looking forward to the meeting as always. It's my favorite meeting of the year. This year, it's going to be in beautiful Montreal, Canada. And so, like Dale alluded to, you are the program committee co-chairs, and wanted to start things off with a question for both of you. You know, how did you think about that overarching vision for ISSCR 2026? How do you connect all the dots in the program itself? And I mean, you know, Dale mentioned some of the things that are going to be of particular interest in this meeting, but were there specific scientific shifts or opportunities or technologies that help shape the program? So maybe Fiona, you could get us started. Just, you know, what is that overarching vision for the meeting this year?
I think one of the biggest themes was really trying to bring together different topics that span everything from the most fundamental discoveries in basic science all the way to translation. And I think a key goal for both of us was really to bring a lot of new voices, as we heard. We really wanted to have early career speakers, mid-career, and more senior scientists who have not spoken before, because there's so much great science out there that we wanted to bring this together. And of course, with all the breakthroughs in technology, we're now able to really look at cells in vivo, follow them, understand lineages, understand how they respond to disease, and go all the way to translation.
Yeah, so I always want to remember the success of foundation model like ChatGPT and LLM. This was a big breakthrough of the year, and the scientific community was also tremendously affected by the success of this. And it's a useful tool, but not only that, I think it also inspired us to study diverse subjects, you know, multi-layer objects. What I mean here is that the success of foundation model basically tells us that the objects like existing in this world is somewhat independent, but there are many fuzzy linkages, and those AI system can distill those fuzzy linkages to derive knowledge. And the same thing is basically happening in biology field for long time. we study diverse organisms, diverse cells, and then find commonalities. So I think we are entering the era that those scientific behaviors or scientists' behaviors that we are taking, finally meet to those AI system. So, this year we highlight AI and also, many new technologies. So I'm pretty excited about that.
Yeah, let's stay on that theme. I was looking at the agenda. We're talking about the speakers here, and there's some real scientific star power, but really nicely woven into this theme of convergence. I mean, the highlight that I'm really looking forward to is the keynote from Aviv Regev, whose work is at the intersection of biology, computation, AI. And really exemplifies that point of emphasis, right? Both convergence, but also this future-facing science. And you just look at all the findings and seminal innovations that have been enabled by the foundational tech that Aviv Regev has developed with others. And it really shows you how much we have to look forward to in the future. Based on the science that is being reported now and this year at the meeting. And just to highlight some of the other big names, we got Azim Sarani, who's given the Anne McLaren Memorial Lecture, also his own keynote. We got Shinya, he's in the game, a late add to the program, Shinya Yamanaka. He's going to be part of the presidential symposium, as well as, just some bombshell plenaries, as well as the concurrence. I also want to come back to that. More than 75% of the invited speakers are first-time speakers. So yeah, Fiona, you emphasize that new voices, right, is a key goal. But just amongst that hit list of innovators and future tech, why don't you guys give us one speaker or one session that you guys are really looking forward to?
So this is really hard because I love all the sessions, but I am a scientist who thinks about cells inside tissues. And so I actually have two favorite sessions. One is the sort of tissue stem cells in physiology and disease, and the other is modeling early development. And I think what's exciting in the modeling early development is we actually really tried this year to also bring in more theoretical approaches and modeling, so not just experimental science and what we can learn from that, and also some ethics perspectives and thinking about diverse populations and how this is very important to think about as we do more translation. And of course, I'm really excited about how the body changes with different physiological states, such as pregnancy. And so Irene Miguel Aliaga is going to be talking about her beautiful work crossing from flies into mice and how reproductive states can change stem cells and affect the whole body. And we also have a big interest in this. But really, and then Aviv Regev, of course, will be speaking in that one. So these are my two favorite sessions, but I'm so excited about all of them.
How about you, Nozomu?
Very hard. But let me take the analogy of AI again. I don't know if I could deliver my observation nicely in English, but the genome is a foundation model, and stem cell is a generative system. So we know generative AI and foundation model, but that's basically what the cell is doing. And therefore, and I feel like we are entering the era of decoding what is encoded in the genome and how such a generative system work. And stem cell is the perfect generative system. So, with that perspective, all of them align, right? So I'm pretty excited about all of them. And especially maybe one thing is, the stem cell community enjoys the beauty of biology, but at the same time, they also translate those knowledge into therapeutics and clinics. So that's another aspect that I am pretty excited about.
And certainly that's going to be very heavily highlighted at this very translationally oriented version of ISSCR 2026. One thing that I'm also, of course, very excited about is the opportunity to catch up with old friends and meet new ones, right? In addition to listening to all these amazing talks. And certainly people have a lot of opportunities to go to different meetings, right? ISSCR is a phenomenal meeting for stem cell biologists, but there's smaller meetings out there as well. And ISSCR, of course, puts together a number of those. But if somebody asks you, it's like, okay, I have the opportunity to go to all of these different meetings. Why should I go to ISSCR 2026 annual meeting in particular? What is it about this meeting that makes me, what is it about this meeting that should draw me in? Fiona, maybe if you want to get started.
I think that in contrast to small meetings, which are also fantastic and very focused, here you're exposed to a great breadth of topics that you normally don't hear about. And I think that's so important also to think about the clinical perspectives. And we also always have a patient advocate or some patient who's involved. And this is very moving and important to understand how our research actually ultimately translates really into helping people. And so I think you come away from this meeting by being exposed to many different areas that you normally don't hear about. And this can spark and inspire your own research, come up with new techniques or ideas. And I think also it's an incredible opportunity to meet people from around the world. We try and build community at these meetings. And through time, you build up wonderful friendships, people you've known for many, many years. It's a great opportunity for early career researchers to meet with and interact with lots of different scientists around the world. And right now, this is what we need in science.
Well, Fiona took all of my answers. But maybe to add to that, we have many big shots, great, fantastic speakers. And if you are students, it's a great opportunity to learn, how they, actually physically exist in the world, and then how they speak and present their work. And also, as you said, 70% of the participants are new to the community. And I always appreciate diversity of different ideas and different ways of thinking. And we should have frictions of those different ideas. And that's always the source of coming up the next step. And I think ISSCR is one of the rare place where we have diversity and lots of frictions, I would say.
Yeah, I mean, that is one of the great points of the meeting is that you have all these, as you said, all these different fields and specialties, but there is it's a safe space, as they say, and I think that's encouraged. Not necessarily friction in terms of conflict, but I think there is people are encouraged to share unpublished data, for example, and to speak about issues that can sometimes be charged and there isn't always global consensus on. And ISSCR has shown a lot of leadership in establishing guidelines to that effect. And I think the meeting in itself is a forum where that same idea plays, right? Where all opinions and hypotheses are welcome. So it is one of the best things about the meeting. And as you also said, it's big, right? It's massive, all these different people. But I want to also say that it's not just a place where you go to be exposed to a wide breadth of knowledge and investigation, but it's also, I think, a place where there's opportunities, as Nozomu just said there. There's a lot of new membership amongst the speakers, but also there's 80 plus oral presentation opportunities that are available to abstract submitters, right? So while we're talking about that, I'll throw out those deadlines for those who are listening, taking notes. February 25th is an abstract deadline. You also get the best registration rates. Also, late-breaking abstracts deadline for posters is April 22nd. But for the trainees, right, who I think I remembered going to these meetings and being just overwhelmed. And you don't have your people necessarily. You're not known. How is the program designed, and how is ISSCR? I know ISSCR has also always placed an emphasis on getting new voices, but how do you create space for interaction, right? How is a program designed to foster these meaningful connections that we all know we've formed at ISSCR, but how do you make that? How do you enable that for trainees for whom I can remember it's not easy?
I mean, one thing I would like to highlight also is the opportunity for early career researchers to design their own session. So we have these spotlight sessions, and that deadline is also February 25th. And so the idea here is that you can create a session of a few speakers, which are then from I think up to five of them will be selected and become part of the program committee. So this is a great way also to get involved, be part of the community, reach out to other scientists, and design your own program. So that was just an aside. In terms of building the community, it's a really important point. There are different places for people to network. There are global hubs where you can come together and interact with each other. There's a big effort also for the early career scientists to have communication channels open that they can interact with each other, find people to go to dinner with. There's going to be this year an all attendee event, which will be a great opportunity also for people just to meet and interact. So this doesn't happen every year, so we're really excited about that. And also really, there's a big emphasis this year on trying to set up physical spaces where we can really interact. And so in some past meetings, it's been harder, but we're really trying to have almost a library of tables where people can go, and even thinking about having some special topics of interest. So if you're interested in a particular theme, you can come together and get to know each other that way.
Yeah. No, I think there's going to be a lot of opportunities for these dialogues across whatever scientific discipline, or even, ethical dialogues as well, in the context of, say, brain organoids, in vitro gamete genesis. I mean, these are things that folks can talk about in these, and hopefully spontaneously as well, in between the meetings, in between the talks in the hallways. That's what the heart of this meeting really is all about. So, before we wrap things up, I of course wanted to ask you a question about Montreal. And Nozomu, maybe I'll ask this to you first, since you're based in Canada, in Vancouver as it is. Going to ISSCR every year gives me an opportunity to explore a new city, explore a new country sometimes, right? I remember experiencing Hong Kong, and that was just an amazing, amazing experience in a part of the world that I've actually never been to. And just the food is something we talked about forever after that meeting. So, Nozomu, what is it about Montreal that makes it such a special place to host ISSCR 2026?
Food.
There you go.
Everyone should enjoy food over there. But I don't know if I am a appropriate person to introduce Montreal. Fiona actually did undergrad in Montreal, no?
Uh-huh.
Yeah, so I'll let her speak.
I did. I studied there, and I love Montreal. It's one of my favorite cities. It has a wonderful feel to it. It's very lively, mix of Europe, mix of North America, really interesting communities and neighborhoods all over, in different parts of the city. It's a beautiful place. You can go up the top of the mountain, or Mount Royal, and then look over the city. And there's just so much life on the streets. So from that perspective, it's really a fabulous place to have a meeting. Lots of cafes, lots of drumming. Yeah.
Drumming. Wow, I wouldn't have guessed. So yeah, I mean, I want to, for two types, two of our targets here. The first time attendee, I think we addressed all the wealth of opportunity there. But for someone who's been to, I don't know, a bunch of meetings, maybe all of them, what's new, different about this 2026 meeting experience that you think would be, a welcome surprise to a long time attendee?
I think we're really trying to provide opportunities for community building in an international world. To me, this is one of the biggest goals, and to just foster these interactions, to provide spaces to talk about ideas, complicated issues that are very, very important. And for example, this year we also have this track, which is really focused and set up by ethics committees, and also by the policy committee and translation. And so talking about very key themes, and people can engage with these. So I think this is the key for me. It's really forming this opportunity to build this community.
Nozomu, what do you think we can expect a bit different?
Yeah, I think we have many new people. Is the very great opportunity for long-term ISSCR member. ISSCR is a very respectful environment, and as a co-chair, I would ask the ISSCR members to keep being respectful and keep welcome new students and new members to the community, and also get inspired. Another thing, to students. When they meet speakers and big shots in the conference, they look probably scary a little bit, but we are all lowering our eye level, so please don't hesitate to interact with them. We are all super nice people, so.
Yeah, just a quick follow-up on that. It just got me thinking, because we have this another recording scheduled with Shubin Cheng and Hans Clevers. And part of that discussion is about standardization and how the ISSCR is really placing a huge emphasis, right? We got another one of these consortiums to try and integrate the industry and academic science, right? So can we just talk about welcoming new people? And I feel like, if I'm honest, it's all us eggheads there, right? We're all there listening to each other, clapping each other on the back, and it's all awesome. Don't get me wrong. But I feel like maybe there is not traditionally, while they've always been welcome, the industrial partners, which are increasingly critical to translating our vision, right? It's not that they haven't been welcome, but they haven't been really present to the degree that their stakeholdership would merit. Anything that we're doing there to make this a meeting that's more aligned with industry? I know a lot of trainees also are looking in that direction, not as an alternative to academia, but as an additional pathway. So I just wonder how maybe the ISSCR is evolving a bit to incorporate that industrial component to annual meeting. Any thoughts there?
Yeah, I think compared to other life science disciplines, stem cell biology is very close to industry and therapeutic translation. This is a pretty unique aspect of stem cell biology. Yeah, as you said, everyone is facing to clinics and therapeutics. Although researchers and students are curiosity-driven people, it's also super exciting that our creativity can easily translate it into, not easily probably, but translated into clinical settings. And that's a great aspect to think always.
I think one thing that I wanted to bring up is that these regulatory discussions are actually actively discussed at the meeting as well. So in this ethics, translation policy, and impact track, many of these regulatory discussions with different countries will be brought up, and including with the FDA and Japanese and UK and European agencies. So as we talk about industry, there is that realm that's being brought up as well.
Yeah, I mean, it's inevitable reality of the meeting, and I think it's going to happen. I think there's going to be more representation from industry, combining with academia as these stem cell derived products move towards patients, as they approach FDA and regulatory approval. I mean, I think these are things that are going to happen, but inevitably these things take time. So wrapping things up here. If you had to really summarize ISSCR 2026, which is just happening in a few months now, if you had to summarize it in one sentence for our listeners, what would that sentence be? Fiona, if you want to start.
Meeting place for great ideas and inspiration.
Love it. Nozomu?
The new era of biology will happen from here.
Powerful.
That is potent. I love this meeting. I mean, this is kind of silly with our bias. We all can't wait. But I mean, for those listening who are skeptical, maybe cynical, you gotta understand. You're gonna come away from this experience. You'll have some cafes and some drum circles under your belt. That goes without saying. But also, you will be inspired, right? To incorporate the other single sentence, it is the future of science that every year begins with that meeting. The future always begins with today. But that is a series of days of spectacle, inspiration, and innovation, I think, that it really stands alone. And we just can't wait. And I hope we've made our case here for all of you to join us. Send in an abstract. Fiona Nozomu, thank you so much for talking with us today. And any parting thoughts or anything else you want to share, by all means.
Thank you.
Thank you. Look forward to seeing you there.
All right, gang, that brings us to the end of this episode, our pre-ISSCR show. Can't wait for that meeting. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. If you enjoyed this episode, please feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or via email at info@STEMCellPodcast.com with feedback or to suggest guests. Next stop, Montreal. Viva les stem cells!